Hey, hi, Year 12. Welcome to another lesson. We're going to be covering one of the most important parts of this topic now, which is meiosis. Now, meiosis is absolutely essential for life of our kind. It's not essential for all life, it's absolutely essential for sexual reproduction. And so we're going to be studying this in a lot of detail. Now, before we get started, I wanted to say this is quite a long lesson. I wasn't quite sure if to split it into one or two. I'm not really quite certain for myself how long this will take on a video. Now, if this is taking you more than 50 minutes, can you please bear in mind that the next lesson, which is on non-disjunction events, is going to be very short, and so that if this is taking you more than 50 minutes an hour, you should stop, put down the video, and come back to it next lesson, and then you can do the end of this lesson and non-disjunction events in 50 minutes, I'm fairly sure. So don't spend all afternoon on this if this does happen to take me an hour or so. Okay, so we're going to start off with the starter, which is about meiosis, and you did some meiosis in year 11. Now, I'm not quite certain whether you guys would have been in school for this bit and how much you remember of it, but I'd like you to write down either a list of notes, bullet points, let's say, or a spider diagram if you're a diagram person. What do you already know about meiosis? Now, it says to swap with your partner. That's a sad relic of the old days. Obviously, don't swap it with anybody, but just have a think. What do you already know about meiosis? Spend a couple of minutes thinking about it, and pause me. Okay, so meiosis. Incidentally, meiosis is the title. We're going to be going into a lot of detail, and I'll go over the basic GCSE you've hopefully just written before we start. Meiosis is how we're going to be creating gametes, sperm and egg, and we're going to go over it in a huge amount of detail. But today we're going to start off by explaining the different outcomes of mitosis and meiosis. That's quite GCSE-ish. You should already know that, but we'll go over it. Explain how meiosis results in variation. That is very A-level. We'll go into a huge amount of detail on that. Complete diagram showing the chromosome content. It's going to take some time. You're going to need probably something round, like the bottom of a cup or something, because we're going to draw in a lot of cells. And then recognizing where meiosis occurs and how random fertilization increases genetic variation. Now, there's a lot there, as I say. So let's get on with it. Now, meiosis at GCSE, all we needed to know was that you form 4 haploid cells, which are called gametes. Now, the word haploid, we remember from GCSE, means it's got half the normal amount of DNA. And that's ridiculously important, because when it fuses with another haploid gamete, let's say the sperm and the egg in humans, or pollen and egg in plants, you want to create a cell with a normal amount of DNA. So we're going to have half the amount in the egg, half the amount in the sperm, they're going to fuse together, and we're going to have a normal amount. So, if you don't know this already, if you don't know the GCSE, please write this down, because it's very important that you already know this. The only other word you might not remember is the word zygote there. It's the last word on the slide. Zygote just means a cell that is formed from the fusion of 2 gametes. A zygote. Always good for Scrabble, at the very least. So, we're going to move on. Pause me if you haven't got those notes down, if you need them, if they were not in your starter notes. I'm going to start off actually by talking about mitosis, because one of the key things we have to understand is the difference between mitosis and meiosis, and so we're going to start off making sure everyone remembers mitosis. So, have a look at that. There's a question there, and it's 1 mark to start with, and we're going to scroll on to the next little bit. So, A, B, C, D, and E, anaphase, interphase, metaphase, prophase, telophase. Write one of the letters to complete the following statement. Chromosomes line up on the equator of the mitotic spindle. So, in which of those do the chromosomes line up on the equator? Hopefully you wrote C, metaphase. Metaphase is where chromosomes line up on the equator. Now, this is a little bit more complicated. It's a bit more of a, a bit more of a long question. It's only 2 marks. Have a quick look. Have a quick look. And pause me for a couple of minutes if it's worth. Right. So, we're talking here about inhibitors causing the development of monopolar spindles in mitosis, and it gives you a little diagram showing what that is. This is a classic kind of question. It's a fantastic question because it refers to some experiment. It's not just saying, what do spindle fibers do? It's giving you something interesting, like a kinesin inhibitor, and asking you, why would this affect things? Which is basically the same question. The question is, what do spindle fibers do? So to suggest why this development of a monopolar mitotic spindle would prevent successful mitosis, we're looking at, basically, originally the spindle fibers should be pulling half the DNA to each side of the cell. But what will happen here is all of the DNA will end up in one cell, which will have too much DNA, and no DNA will end up in the other cell, which will have no DNA and will die. So you'll end up with one cell rather than two, with too much DNA. Okay? Fine. Right. Now this is an easier question, and then we will look through the proper mark scheme for you. So the figure below shows how the amount of DNA per cell changed during interphase of meiosis. Oh, I think this is fairly GCSE, to be fair. Have a go. Okay. So we're looking here, this is a GCSE-ish question. So we're looking here and we're saying that between F and G, the amount of DNA is halved because we've created a haploid gamete because the DNA is split in half. Now that's about all we have to do at this point. We have to answer that question a bit fuller at the end of this lesson. And then the final question we're going to do to start with is this one. And this is basically being able to identify which stages are which. So what order do these go in? And then explain that graph. This is not a proper A-level question, obviously. Fine. So, the order. So we're looking at B, we can't actually see any chromosomes, because that is before mitosis happens. All the chromosomes are completely well, not completely uncoiled, are less coiled, and so they're kind of forming a blob. B is first. Okay. Then you've got A. A is actually metaphase here. Then you've got E, which is anaphase. You've got C, which is towards the end of telophase, beginning of cytokinesis. No, it's not. I apologize. D is towards the end of telophase and cytokinesis, and then C is after cytokinesis has occurred. So the answer should be B, A, E, D, C. Fine. And explain the graph. Well, in G1, we're just growing the amount of organelles. The DNA doesn't change. S phase is DNA synthesis, so we're going to double the amount of DNA. In G2, again, we're just growing loads more organelles, so the DNA doesn't change. Mitosis. At the beginning of mitosis, the DNA will just be lining up in the middle of the cell, getting prepared to be pulled apart, and at the end of mitosis, the cell will split in two and we'll go back to a normal amount of DNA again. Now, those questions came from all over the place. They're all different kinds of questions. If you at least followed most of them, then you probably got mitosis fine. If you don't know prophase, metaphase, anaphase, telophase, cytokinesis, what happens in each one, you will not be able to do today's lesson. So you need to then pause this video, go away, and make sure you learn from mitosis what happens in prophase, metaphase, anaphase, and telophase. Because the exact same things happen in prophase, metaphase, anaphase, telophase in meiosis with a little bit more difficulty. So it's actually a little bit more difficult. So you can't start unless you know it. Go away, get your textbook out, prophase, metaphase, anaphase, telophase, learn what each one does. Okay, so there we go. Now, this is basically what we just talked about. We already wrote these notes, so don't write them again. We're looking at creating haploid gametes, sperm and egg. Now, a general overview looks like this, and again, this is still GCSE. So we're imagining there is a cell with only one chromosome pair. And obviously, we have 23, and so we would have 46 total chromosomes. This only has one chromosome pair. Still, between here and here, we have an S phase. So, before meiosis occurs, we still double up the amount of DNA. So the first 3 stages of meiosis, creating gametes, are exactly the same as the first 3 stages of the mitosis process. You still do the rest of the cell cycle. G1, S, G2 still happen. So from a normal adult cell, the first stage is G1, we're going to get more organelles, going to grow. S, we're going to double the amount of DNA we have. G2, continued growth, more organelles. And then we have this cell just below here, the second cell. Now that cell now has twice as much DNA as it should. The top cell has the right amount of DNA, the second cell has twice as much DNA as it should. And this is where we change. In mitosis, what would happen is, this blue cross would be split into 2 blue chromosomes. This yellow cross would be split into 2 yellow chromosomes, and we'd end up with a blue and a yellow in each of these 2 cells. And they would be identical. So at the end, after this second cell, we would just have a cell with a blue and a yellow and another cell with a blue and a yellow. And you can see in meiosis that isn't what happens. In meiosis, the first step is we divide these pairs of chromosomes up. So in each chromosome pair, we send one part of that pair to one cell, one part of that pair to another cell. So you have 46 chromosomes, 23 pairs. At the end of meiosis 1, and we'll go into a little more detail in a minute, at the end of meiosis 1, 23 of those chromosomes will be over here, 1 from each pair, and 23 chromosomes would be in another cell. So you'll end up with 2 cells, each with 23 chromosomes. So instead of having pairs, you now only have single chromosomes. 1 from chromosome pair 1, 1 from chromosome pair 2, all the way down to 23. We can't stop there though. We do now have half the amount of chromosomes we should. We've only got 1 of each pair in each cell, but each of those chromosomes still has twice as much DNA as it should. You can see it's a cross. So after meiosis 1, you've got this cross. You can't leave it like that. That's not what cells should be like. DNA won't function like that. So we then have to split those chromosomes up in a process very much like mitosis, and we're going to end up with 2 cells from each of these 2 cells, each with the right kind of DNA. And if you compare the end of meiosis 2 at the bottom, these 4 cells, to the adult cell at the top, you can see each one has half the amount of DNA, and each one only has 1 of the pair, 1 of the chromosomes. Okay. Now, if you don't understand that, if you haven't followed that, A, go back and watch me again. It's 2 minutes of me talking. Or B, there's a little link at the bottom there. Now, I will try and remember to put this in the video link. Sometimes I remember, sometimes I don't. If I don't remember, just type it in. It's a few numbers, it's annoying, but it's quite a good animation, which basically explains what I've just said in animated form. Fine. So, let's talk about meiosis 1. So, meiosis 1 happens after G1, S, and G2. So before this happens, we've doubled up our amount of DNA, we've duplicated our DNA, we've grown the cell massive, and we've got large numbers of organelles in there, just ready for division. And at that point, you could do mitosis or you could do meiosis. Up to that point, it's the same. Once we've made the decision to go down the meiosis front, once we've, in the testes or the ovaries, made that decision, things start to change. So, we still have prophase, metaphase, anaphase, and telophase. Now, prophase 1 is very similar to prophase in mitosis. The chromosomes condense, the nuclear envelope disappears, and you're left with things that look like this. And at this point, you have the right number of chromosomes, that's all fine. So, In prophase 1, those things happen. But at the end of prophase 1, we have an extra step. Okay, the first difference between mitosis and meiosis. So if you can write down that there, in meiosis 1, several similar processes, etc. But there's one more process that happens, and that's called crossing over. Now, this is a picture of crossing over. I'd like you to pause me, have a look at that picture, maybe draw yourself a very similar diagram, just a simple one, and then can you write down what you can see happening there? I'll explain it in a second, but from the diagram, I'm trying to get you to think about it rather than just listen to me. What can you see happening there? And after that, how will that affect genetic diversity? So pause me, please do have a go at this task, don't just wait for me to tell you. Okay, so crossing over, what's happening here is in each of the pairs of homologous chromosomes, so in every pair, they will kind of sidle up to each other. They'll wrap their chromatid arms around each other, and remember, each one of these bits of the cross is a chromatid, so you've effectively got two chromatids here. They'll wrap their chromatid arms around each other. They wrap around each other, they break off, and then they reattach on the other chromosome. So a little bit of this blue one will end up on the red one, a little bit of this red one will end up on the blue one. The chromosome pairs twist around each other, they break, and the broken end attaches to the new chromosome. And this can happen thousands of times on each chromosome. So what you end up with is, at the end of crossing over, each of these chromosomes won't be red or blue anymore. There'll be a complete mishmash and mix-up of the two. Because bits of the red have snapped off and attached to the blue, bits of the blue have snapped off and attached to the red. And what this does is it means you get a lot more genetic diversity. Because what's happening is, rather than just, let's say, when you have children, they get your dad's chromosome 1 and your mum's chromosome 2 and your dad's chromosome 3 or whatever, they're getting a complete random mixture of the chromosomes that you got from your mum and your dad. Because they're all twisted up and broken apart. And that leads to a lot of genetic diversity. Because if you had two children, let's say, one of your children will get a chromosome 1 that has a lot of blue and quite a little bit of red. The other child might get an enormous amount of red and only a tiny amount of blue. But it would be completely random. This is at the end of prophase 1. So we've condensed the chromosomes, we've dissolved the nuclear envelope, then we wrap around each other, we snap off. Crossing over. Add anything to your notes that you need to from this page, and then we'll move on. So let's move on to metaphase. Now, in metaphase of mitosis, what happens? Can you remember? Hopefully you're thinking about all the chromosomes lining up down the equator, one on top of the other in a big old line, and the spindle fibers attaching to them. What difference can you see in that image of metaphase 1 in meiosis? Well, they're not all lined up one above the other like in mitosis. In meiosis, the chromosomes line up in their pairs. They line up 2 by 2. So the two chromosome 1s will line up next to each other, the two chromosomes 2 will line up next to each other, all the way down to chromosome 23, though not necessarily in order, but you will end up in pairs. So the spindle fibers will attach still, but the spindle fibers on one equator of the cell, on one pole of the cell, will attach to one of the pair, and the spindle fibers on the other pole of the cell will attach to the other one of the pair. So when the spindle fibers reel in, those chromosome pairs are going to be ripped apart. So half the chromosomes will go in one direction and half the chromosomes will go in the other direction. And this is where we divide up the DNA and we have half the usual amount of chromosomes by the end of this. For metaphase, let's not get ahead of ourselves, they're just lining up. They're lining up in pairs, and the spindle fibres are attaching on. Now, something happens here called independent assortment. Now, independent assortment means that it is completely random on which side of the cell your maternal and your paternal chromosomes will line up. Let me explain. So if we look at our cell in our little diagram, it's my left, I can never remember if it's your right or not. On my left, at the top, there's a blue, but on the second pair, the blue has ended up on the right. They're on opposite sides. Now this bottom blue chromosome could have ended up on the other side. It could have ended up directly underneath the other blue chromosome. The top one could also have ended up on the other side. It's random. Picture me taking our entire class and lining you up in pairs, and let's say there were 50% men and 50% women. Okay? If I didn't tell you that all the men had to be on the left and all the women had to be on the right, it would be fairly random. So maybe in the first pair, the girl ends up on the left and the boy ends up on the right. The second pair is the same, but the third pair is the opposite. It doesn't really matter, but it's random. If I then said to you, right, all the people who are on the left side of the line go to one side of the classroom, all the people on the right side of the line go to the other side of the classroom, I would end up with two groups of people with a random selection of girls and boys in it. If I did this to 1 billion classes, occasionally I'd end up with a class where all the girls are on one side and all the boys are on the other. But usually it's a mixture. And it's the same in your cells. When you're creating your gametes, you are making these gametes from your cells. Your cells contain half the chromosomes you got from your dad and half the chromosomes you got from your mum. So when you are making your sperm or egg, you are creating a cell which contains your mum and dad's chromosomes. But it's not that in all of the sperms, half of them will have your mum's chromosomes and half of them will have your dad's chromosomes. It's a random mixture because of independent assortment. Because the chromosomes line up in random places. So sometimes half of them will be your mum's and half of them will be your dad's on each side. Sometimes it will be that one of the cells you create has mostly your mum's chromosomes and one of them contains mostly your dad's chromosomes. But it's random. And every time you create sperm or eggs, it's entirely random. So every one of the 250 million sperm released during ejaculation contains a random selection, a different selection of your mum's and your dad's chromosomes if you're male. And every egg released, if you're female, contains a random selection of your mum's and your dad's chromosomes because of the way they line up during metaphase. And this is called, and I'll flip back a bit so you have the spelling, it's how you expect, independent assortment or independent segregation. And in actual fact, to be fair, I'm thinking that in the mark schemes it's usually called independent segregation. You can call it either, but let's call it independent segregation now I think about it. So that is there. But that's in metaphase 1. Okay. You'll be pleased to know we've got through the hard bit of meiosis. Okay, prophase 1 and metaphase 1 are the hard bits. Anaphase and telophase 1 are very similar to normal anaphase and telophase. Normal. To mitotic anaphase and telophase. So in anaphase of mitosis, what happened? Well, the spindle fibers kind of reel themselves in, the chromosomes are broken apart, and one of each chromatid goes in each direction. In this case, it's slightly different because the chromosomes lined up in pairs, one of each pair goes in the opposite directions. So in your pair of chromosome 1s, one will be reeled to one side, one will be reeled to the other side. And there's your notes. At the end of this, you have halved your amount of genetic material. You only have one copy of each chromosome. Now sometimes people get confused because these are still crosses. But because they're still attached together as crosses, they're technically one chromosome. So technically, by the end of this stage, if you're a human, each cell will have 23 chromosomes. They're big chromosomes, but each cell will only have 23 because half have gone one way, half have gone the other. Okay, let's scroll down to telophase. Telophase is pretty much exactly the same. The nuclear envelope reforms, chromosomes often decondense. The chromosomes decondense. People ask me, why do they decondense when we're about to go into another stage? It's a layover from mitosis. It's just a thing that cells do. It's not really necessary. Right. That's the end of my meiosis 1. Meiosis 1 is by far the harder of the two to remember. So from the top, very quickly, you've got meiosis 1, you've got prophase 1, where the chromosomes condense, the nucleus disappears, centrioles form, and crossing over happens. Metaphase 1, the chromosomes line up in their pairs. And this is what causes you to turn to a gamete and not just two identical cells. Anaphase 1, the chromosome pairs are split up, so one of each pair goes to each side of the cell, each pole of the cell. Telophase 1, same as normal, nucleus reforms, chromosomes decondense. Now, if you have a look at this cell has, and then, to be fair, we have cytokinesis 1, to be fair. Cytokinesis 1 at the end of this, so you're going to split this cell in half. Mustn't forget that. Cytokinesis 1 is exactly the same as cytokinesis, it's just splitting the cytoplasm and the cell membrane in half. Okay. Now, there's 5 questions here, but before we do them, I just want to kind of go over this. So once we have cytokinesis here, I want you to kind of imagine there's a line down the middle. You can see that if this cell had 2 pairs to start with, which it did, we now have 2 cells, each with 1 of each pair. So this cell is haploid. There were 4 chromosomes in the cell, there are now 2 in each cell. But each of these chromosomes has too much DNA. They should just be single straight lines. They are actually crosses. They have twice as much DNA as they should. We need to fix that. Now, that's meiosis 2's job. Before we move on to meiosis 2, to make sure you've got it, there's 5 questions there. You probably have these notes down, you should, so I don't expect you to write out these in full. I just want the answers, please. And if you want to do it at the back of a book or on the other side of a piece of paper, you can. Pause me. Okay. So, crossing over. Crossing over is where the chromatid arms of two homologous chromosomes, or of a homologous pair, wrap around each other. The arms then break off and reattach. When does crossing over occur? At the end of prophase 1. In actual fact, the process is completed in metaphase and anaphase 1, but at the end of prophase 1 is when the actual crossing over bit happens. Why is crossing over good for sexual reproduction? Because it massively increases genetic variation. We can talk about how many possibilities there are of how many possible combinations of 46 chromosomes you can make, but it is relatively finite. With crossing over, you can have almost infinite possibilities, and that's why almost nobody looks identical to anybody else. What is the difference from metaphase and anaphase between mitosis and meiosis 1? The difference is the chromosomes line up in pairs, not on their own, so that during anaphase, the homologous pairs are split up. So you have 1 from each pair going in each direction. Now the final cells, haploid or diploid, they are haploid cells. Okay, meiosis 2 is, to be honest, mitosis. It's identical to mitosis in pretty much every way, except that we are dividing a haploid cell. So what happens is, for each of these two cells we created at the end of meiosis 1, the chromosomes decondense, the nuclear envelope disappears, they line up one above the other in the center of the cell, the spindle fibers attach, the chromatids are broken apart, and you end up with 2 cells for each cell. Now, crossing over doesn't occur in this position. There is no crossing over happening here. I will fly through this, but in all honesty, it's mitosis. So meiosis 2 is pretty much mitosis. At your level, at least. When you get onto degree, it's a little bit different, but not at A level. So prophase 2, chromosomes recondense, nuclear envelopes break down again. I'll leave that there for 5 seconds so you can pause me. Metaphase 2, the chromosomes line up down the equator, the centrioles attach to spindle fibers at the centromers. Anaphase 2, the chromosomes are split and one half of each migrate to the balls of the cell. And telophase and cytokinesis. The cytoplasm splits to form 4 haploid daughter cells. You decondense your DNA, you reform your nucleus, and now you've got 4 haploid daughter cells, each of which is slightly non-identical. Now, I'm not going to spend long on meiosis 2. If you don't get meiosis 2, go back and learn mitosis, it's the same. A question you will be asked an enormous amount is how meiosis produces variation. So have a think, what parts of meiosis did we just say produce variation? And the answers are 1. Independent segregation. Because the chromosomes line up completely randomly, sometimes you end up with more of your maternal chromosomes, sometimes more of your paternal chromosomes, but every single gamete will be different. Not every single, but there are lots and lots of combinations of different gametes. Lots of variation. The other step, crossing over. Crossing over is the great creator of variation. Now, it depends if we're talking about meiosis. I don't really include this as part of meiosis, but if it's asking you how sexual reproduction causes variation, which is a similar question asked a lot, you want to talk about random fertilization. Even with all of the variation we're talking about, there are only so many possible combinations of DNA. So, every time a male and female have sex, there will be some similar sperm released, and there's a fairish chance of similar eggs being released. They're a lot less common than similar sperm, because there's a lot more sperm. But every time, it's completely random which sperm will get there. And it's also completely random which egg will be released. That's a lie. And it's also genetically completely random. So there is a decision-making process within the ovary as to which egg will be released, but it's random which egg, what the DNA within that egg will be. So, even if you released exactly the same 250 million sperm twice, a different sperm would fertilize it each time. And so the zygote would have different DNA by the end. That's called random fertilization, and again that increases variation. Fine. Now, can you draw this table? I'm going to give you some facts. It's going to be quite straightforward in all honesty. This is kind of a general overview. But just draw me this T table, and I'll give you some facts, and I want you to write in which fact goes where. So, meiosis, mitosis, there's going to be 16 facts, I think, 8 to start with. There you are. So pause me and just put them in the right place. Right, and then the second possible ones, I think there might be some overlap there. Right, so if you haven't already, pause me, do the task, and then I'll go through the answers now. So, half the number of chromosomes in cells, meiosis. Enables a species to adapt to a changing environment or to colonize new environments, meiosis. Variation leads to adaptation. If every single person has different DNA, some will be better adapted, some will be worse, so that leads to adaptation over time, due to natural selection. Produces cells with a diploid number of chromosomes, mitosis. Enables a species to colonize an area quickly, mitosis. That's bacteria. When out of control, a cancerous tumor may result, mitosis. Important for the repair of damaged tissues, mitosis. The original parent cell become two daughter cells, that's mitosis. 4 daughter cells are made, meiosis. Produces new genetic combinations, is meiosis. Daughter cells contain half the number of chromosomes, is meiosis. Cell division by reduction, meiosis. Daughter cells have the same number of chromosomes as the parent cell, mitosis. The daughter cells are copies of the parent cell, mitosis. Happens only in gamete-producing organs, meiosis. Now, perhaps we should have done this at the beginning of the lesson, because that's quite a lot simpler. You will be asked differences between mitosis and meiosis, and you can just spurt these ones, that's absolutely fine. Okay, let's do some questions on this. Oh, sorry, there is one more thing. A last thing you're expected to know. A little bit of maths. Basically, they could, and I've never seen a question on this in the A-level, but technically they could ask you to calculate the possible number of combinations of chromosomes that can arise from a independent segregation. So if you've got n numbers of chromosomes, the way you calculate the possible number of combinations is 2 to the power n. So if you only have one pair of chromosomes, there is only 2 types of cell you can make. If you have one pair of chromosomes, one chromosome will go one way, one the other way, and that will make you 2 cells, and those are the only 2 combinations. If you have 2 sets of chromosomes, there are 4 sets of cells you could make. So, and if I have 4 chromosomes, it will be 16 combinations, and so on. So, I'm going to give you some questions on this, make sure you can do it. It's such simple math, I don't anticipate anyone not being able to do it, but remembering this can be difficult for some people. 2 to the power n, you just need to know that. It's unlikely to come up. But make sure you can do these questions. If you can, then you will be able to do these questions. It's not difficult. So just pause me and do these. Okay, I will be honest with you, gents, ladies, I would usually have a calculator in class and be doing it while you do it. So I'll give you the sum you should have done, and I would imagine that you'd get the answer. So, right, by random independent segregation, if a fly has 36 pairs of chromosomes, the answer should be 2 to the power 36. You can work out what that number is. A gamete of a monkey that has 16 chromosomes, that's 8 pairs, so it's 2 to the power 8. Be careful of that. An adult cell has 64 chromosomes, that's 2 to the power 32. And a human creates a gamete, that's 2 to the power 23, because we have 23 pairs. And why are there far more combinations of genes that are created by meiosis? Well, because of crossing over. Okay. Right, now let's do some different questions, some perhaps more interesting questions. Right, I'm going to flick through these for you. I'd like you just to complete the question, and we'll go through the mark scheme once we've done all of the different parts of the question. So complete this one, it's a 2 marker. Fine. And then we have this one. And that incidentally is a 2 marker. I'm assuming you're pausing me each time here. We have that one, part C. And I apologise, the marks aren't coming up, that is a 1 marker. And then we have part D, which is a 2 marker. And then we'll go through the mark scheme. Okay, so we had firstly A, which was describe and explain the appearance of one of the chromosomes in cell X. So, chromosome is formed of 2 chromatids, that's why it's an X. Because DNA replication has occurred, and sister chromatids are held together by a centromere. Now, this is something sometimes people struggle with, is the difference between a chromatid and a chromosome. A chromosome is just a big bit of DNA. Now, chromosomes usually just look like long, straight lines of DNA. After DNA replication, though, we've copied that long, straight piece of DNA, and now we have 2 long, straight pieces of DNA. They are attached together, though, and so they are technically just 1 bit of DNA as a cross. So that is 1 chromosome. The original straight line was a chromosome. Now we just have a much bigger chromosome, a double chromosome, if you like. And each of the arms in that double chromosome is a chromatid. A chromatid is 1/2 of your little X. When you break them apart, we now have 2 chromosomes because they are now separate bits of DNA. A chromosome is just a big bit of DNA. So, chromosome formed of 2 chromatids, that's why it's cross. DNA replication has occurred, that's why we have 2 rather than just 1. And sister chromatids held together by a centromere. Fine. Any 2 of those. Then we have B, which is describe what's happened during division 1. Well, the chromosomes are in homologous pairs, and 1 of each of those has gone into the daughter cell. So we've said they've lined up in pairs, and 1 of each has gone into each daughter cell. If you went into detail about prophase, being crossing over, and independent assortment, no issue with that at all. Because it's only worth 2 marks, you're not getting any marks for that. You should always write as much as you can think of, as long as you have time. And you've got separation of sister chromatids, or division of the centromere, is what happens during division 2 but not division 1, i.e. we're breaking apart the 2 chromatids, the X, into 2 chromosomes. That doesn't happen in division 1, as you can clearly see. And then D, which was name 2 ways in which we reduce genetic variation. I have to say that question comes up all the time. You've got independent segregation, or assortment, and crossing over. This word chiasmata, another good Scrabble word, incidentally, but the chiasmata just is the bit where the chromatids wrap around each other. The connection they have there is called a chiasma, and many of those are called chiasmata. So you can describe crossing over as the formation of chiasmata. You don't need to know that word for A-level, but it's a good word. Right, I seem to think, I don't have a time of how long this has taken me. Oh yes I do, it's taken me 43 minutes. So it's as I predicted, this will have taken you longer than 50 minutes. I hope you didn't spend too long on it. And I hope you split it over the 2 lessons, because non-disjunction events will be quite a short one. But I hope you've got an understanding of meiosis now. This is an extremely difficult lesson. Not because the actual pieces of information are difficult, but because there's so many little concepts to get your head around. The chromosome versus chromatid, the different meiosis 1 and meiosis 2, prophase, metaphase, anaphase, telophase, and all of that. It's worth going over this a few times. Not necessarily watching me a few times, I think there's only so long you can stare at me, but your textbook, online resources, other videos, exam questions, just go over it until you feel very confident that you understand meiosis, because it does come up. Hopefully it's been useful to you. If you have any questions, I'm on Teams during our lessons, and I'm thinking of starting up a little live lesson during this one because I think people will struggle. And I'm looking forward to seeing you all whenever I get to see you all. Oh, last thing, I apologise. Can you please upload this question, just this question, to Teams? Now from now on I don't want all your notes uploaded most lessons, but I do want this question uploaded. You probably already marked it. Fantastic. I basically want to see how people are doing with these questions, because if people aren't getting it, we need to go back over it. Okay, so just take a picture, good or bad, upload it, and I will have a look at it. Okay. Have a great day. Look forward to seeing you whenever I can.